So we talked about the terminology of different mutations, and then let's talk about DNA replication and what if there is an error made during the process, how that would affect the mutation. So yes, if you look at this diagram, so yeah, so there is a original DNA molecule there. So first round replication, let's say there was a missing cooperation, so there was the original sequence there, CGA GT was the original sequence, right? But let's say through a mistake of maybe DNA polymerase, so this is semi-conjugative replication, so this will be used as a template, and then this will be used as template as well, right? So although DNA polymerase read A there on the template DNA, it should have inserted T on the newly made strand, but by accident it added G instead of T. Okay, and then that was not corrected for some reason. Okay, then let's see what's going to happen. So the bottom strand, there was no mistake made, so it was the same DNA as the original. So then what happens? So let's say this DNA is used again as a template, so semi-conjugatively. So this, should be fine. It's gonna go, but what if this DNA strand is used as a template? Then when it's replicated, see, now there's a completely new base pairing that's inserted there. So that will be the consequence of a mistake made during the DNA replication. Okay, understand that. And then another terms that are used a lot in describing mutagenic condition. So there are certain genes that are very important in maybe inducing the mutations for other genes. Okay, think about that. So the genes that are involved in maybe repressing mutations. Okay, think about if there is a mutation, okay, causing it to be non-functional, the mutation will increase the frequency of mutation on other genes. What does that mean? Think about, yes, let's say there is a mutation made on maybe DNA polymerase itself. Okay, so of course DNA polymerase enzyme is a protein, right? So what if that gene codes for that DNA polymerase was mutated? So of course that's going to affect the structure of the DNA polymerase and then maybe the function of it. Okay, let's say among different parts of the DNA polymerase, it was affecting especially the exonuclease activity. So exonuclease activity is what? That is the part responsible for proofreading of its polymerase action. So since it could not do the proofreading function no longer, so what's going to affect is probably DNA polymerase will make a lot more mistakes, and then because the mistakes are not being proofread and fixed through this activity, right? So the consequence of this mutation on the DNA polymerase encoding gene is what? When this polymerase is replicating other chromosomal DNA, and it's going to generate so many mutations, right? So under this condition, that mutation can happen on DNA polymerase coding gene, it will be considered as a mutator gene because it induces a lot more mutations on other genes. Does it make sense? So not only the DNA polymerase enzyme coding gene will do that, also we'll talk about other enzymes that are involved in DNA repair. If there are mutations made on those genes that are coding those DNA repair process, repair enzymes, then of course they will be considered as mutagen because repair will not be done. Okay. Another important term here, so reversion. Okay, reversion means back mutation or reverse mutation. Okay, what does that mean? So let's say there's a mutation made on one gene. Okay, somehow there will be a secondary mutation made, maybe on that gene on the same spot where the mutation was made. So through the secondary mutation, if the primary mutation was mutated back to the wild type, the original sequence, then we call that a reversion. Okay, so reversion may happen under the very mutagenic condition because there are multiple mutations made, especially on that same gene, maybe the same spot. Okay, mathematically, what is the chance? Probably that will be very slim, right? So that may be only possible if the condition is really, really bad. There are so many mutations happening so high, in high frequency. Okay, then you may get true reversion because there are multiple mutations made on so many spots, and along those, on the same spot where the primary mutation was made, and there's a secondary mutation made, so that, primary mutation is mutated back to wild type. That's what true reversion is, right? And another possibility is there's something called pseudo reversion. Pseudo is kind of fake, right? What does that mean? So yes, the secondary mutation is made, okay, somehow, although the secondary mutation is made not on the same spot where the primary mutation is made, but somehow the secondary mutation that is made somewhere else, somehow that secondary mutation can suppress the phenotype change of the primary mutation. Okay, so then we can consider that as the pseudo reversion, meaning it's a fake reversion because the secondary mutation was not made on the primary mutation site, so the primary mutated sequence was not reverted back. But because of the secondary mutation which was made somewhere else, somehow the secondary mutation can suppress the phenotype of the primary mutation, so that we will consider as a pseudo reversion. Okay, how is that possible? We'll think about the possibility in our next slide, but understand the terminology. So in that case, we not only call that pseudo reversion, we can also consider it as a suppression mutation because the secondary mutation is suppressing the primary mutation. Okay, so think about that. So the suppressor mutation in this case can be made not on the same spot where the primary mutation was made, but it could be still made on the same gene as within the same gene somewhere else within the same gene as the primary mutation. Okay, or it could be made totally somewhere else on a different gene. Okay, so it depends on which condition it is. If it is made within the same gene, we're going to consider it intragenic suppression because it was made on the same gene. But if it was made on a different gene, we're going to consider that as an intergenic suppression. So understand the difference in the terminology. Intra, probably within the same. Inter means probably between the different genes. Okay, so understand intragenic suppression versus intergenic suppression. Okay, so I have this slide in the PowerPoint slide, but I don't like this figure because this is totally a messed up figure. I don't know who made it, U.S. National Library of Medicine, but they did it wrong, totally wrong. So anyway, understand nonsense mutation definition. That's all you need to know, but this figure is horribly wrong because if you look at it, there's that DNA sequence, right? And then there's a transcription process that's skipped. Actually, you don't get amino acid codon directly off of DNA sequence because, amino acid is read from your mRNA sequence. So there should be mRNA sequence. Actually, that was the opposite, orientation. But anyway, just understand the meaning of the nonsense mutation here. So if there is a CAG, it's impossible to get that glutamine from CAG DNA codon. Actually, it should be CAG on the RNA. Then you can get, glutamine. Okay. So anyway, let's assume that this is maybe an RNA codon, not the DNA. And if somehow the C is changed to T, and I guess on RNA, in this example, although it's not possible, RNA doesn't have T, but this is awkward, diagram for that reason. But anyway, if that's the case, then, the T is switched to maybe U. UHG, this is horrible. I mean, let's not use this. That's bad. Okay, so let's eliminate that. Sorry about that. So anyway, that was a bad diagram, so I removed that. So let's use better one, this one. Okay. So let's take a look at this. So let's say this is a DNA original sequence. There is a codon AAG there. The opposite strand has TTC there. Okay. So let's say the bottom strand was the coding strand, which is transcribed into this, which has the sequence of AAG on the mRNA. Okay, so AAG codes for what? AAG is a lysine, right? So when it's translated, it becomes lysine there, right there. So that's the normal, gene, which will give you that protein sequence. Okay. N-terminus and C-terminus. See, this is the correct transcription, because you know, the transcription happens anti-parallel, so the orientation is changed. So this is a better, much better figure. Okay, so now you will have a mutation. Okay, so the I guess the mutation was made where? Or maybe here on that spot. So instead, the original was AAG, there now it's changed to TAG, which will make a change on that opposite strand, which is ATC. So if ATC is transcribed, you will have UAG showing up there, UAG codon on your mRNA. Okay, so it's the translation will happen from 5' to 3' N. Yes, that's fine. It's gonna go until that codon, but UAG is what? UAG is one of the stop codons, right? One of the 3 stop codons. So the translation terminates right there. So since the protein is made in the, until very short and very in the middle of it, it's stopped. The translation is not completed, so you're going to get something gibberish, so non-functional, maybe garbage. So this protein is never made. So let's say that is the nonsense mutation. Okay. So then what? Let's say on that condition, you can expect to have a suppression made. Okay, so what is the possible suppression? So there is that original, nonsense mutation there, but the secondary mutation is somehow, there is a gene coding for the tRNA, and particular tRNA, and then if the gene coding for the tRNA is changed, so the tRNA sequence will be changed when it's transcribed, right? So that the reason why these stop codons stopping the translation is, there's no tRNA that has the matching anticodon that can be binding on this codon, okay, inside of the cell. That's why translation is stopping. But somehow now, there's a mutation on the gene coding for tRNA, and somehow through the mutation, the tRNA, which originally was not able to recognize this stop codon, now it can recognize, it can bind, because it has its anticodon sequence that matches up. Okay, and then let's say that tRNA is bringing in the tyrosine amino acid. Okay. So the result is the translation can continue. Okay, so something functional, protein can be still made, okay, through the secondary mutation. Okay, under this condition, then yes, there are 2 mutations, right? Originally this nonsense mutation, and the secondary mutation which was made on the gene codes for the tRNA, and especially that spot where the anticodon sequence is on the tRNA is switched around. So now that tRNA, which is originally recognizing and the codon that brings the tyrosine, now can bring in tyrosine on that stop codon spot, so the translation can continue. So through that, you can still make this protein. Okay, so that's the suppression we are talking about. So the phenotype of the primary mutation is suppressed by the secondary mutation that is made on someone else, other gene, in this case, the gene that codes for tRNA. So it's going to be considered as what? Intergenic suppression, because the suppression is made through a totally different gene that codes for tRNA. Does it make sense? So understand definition of the suppression, especially intergenic suppression in this case. So it's probably the same thing showing up there in more detail. So UAG is a stop codon. It's going to stop the nonsense mutation. Now, the tRNA is showing up there. So the tRNA with the anticodon sequence, maybe AUG, can so this is a gene coding for tRNA. It's showing up there. So they are showing up. So that's the original tRNA sequence. Let's say that the tRNA gene is also mutated, so AUG is changed to maybe AUC. Okay, that's the anticodon sequence of the tRNA. Now this tRNA can come along and bind on that mutated stop codon spot, because they can match up codon on the mRNA and the anticodon on the tRNA. which was G, now it has C. So AUC is recognizing UAG. It's a perfect